Dear students, welcome all of you in this session. I will discuss here about DNA, the deoxyribonucleic acids. The amazing biomolecule which is carrying the genetic information of all the living higher organisms. Now, see the slide. In the left side, you can see the simplified diagrammatic representations of DNA molecule. In the right side, you can see the detailed molecular structure of a DNA molecule. From here, you can see that DNA molecule looks like a twisted ladder. This structure, also known as double helix, where two polynucleotide chains, one is depicted here as green ribbon, another one as the red ribbon. So here, two polynucleotide chains are interwinding each other to form this kind of twisted ladder. The DNA molecule is chemically extremely stable, but the query is why and how? What is the molecular basis of DNA? Now, all of you know that DNA is a type of nucleic acid. The basic building block of DNA is nucleotide, where each nucleotide are composed of three different components. One is sugar, phosphate, and nitrogenous base. The sugar of one nucleotide is linked together. The sugar of one nucleotide is linked together to the phosphate of another nucleotide to form a polynucleotide chain or one single strand. Adenine, guanine, cytosine, and thymine are the four types of nitrogenous bases present in a DNA. Now, in the next level, you can see that these four bases are able to form hydrogen bond in a selective fashion, which is known as complementary base pairing. It means that adenine can form hydrogen bonding only with the thymine, whereas guanine can form hydrogen bonding only with cytosine. Now, the basis of one polynucleotide chain is forming hydrogen bonds with the complementary bases of the opposite strand or another strand. In this way, DNA is forming double helix structure, the phenomenal twisted ladder form. The sequence or order of the nitrogen bases in a DNA molecule is the genetic code of any life form. Dr. Edwin Chargas first observed this possibility of complementary base pairing in during his experiment. He found that on a molar basis, the amount of adenine is always equal to the amount of thymine, and amount of guanine was always equal to the amount of cytosine. These quantitative relations between four bases amount is known as Chargas' rule. This rule provided a key clue which helped Watson and Crick team to deduce the double helical structure of DNA. In 1953, Watson and Crick took advantage of Chargas' rule and X-ray diffraction data of Rosalind Franklin and M. Wilkins, obtained from the DNA fibers, to work out the 3D structure of DNA. The model they devised fit well with all the experimental data available on the composition of DNA molecule and now famous double helix model of DNA. Here, I need to say that the elucidation of structure of DNA was a momentous occasion in biology, leading directly to our present understanding of life. The main features of Watson and Crick's double helix model is summarized here. The DNA molecule consists of two polynucleotide chains wound around each other in a right-handed double helix. Next, the two strands are oriented in opposite direction. That means that they're anti-parallel to each other. The sugar phosphate backbones are on the outsides of the double helix with the bases oriented towards the central axis. The bases in each of the two polynucleotide chains are bonded together by hydrogen bonds. Now you can see that the base pairs are 0.34 nanometer apart in the DNA helix. That means here you can see in this figure that the distance between two consecutive base pairs is 0.34 nanometer. And a complete turn, that means 360-degree turn of the helix takes 3.4 nanometer. From this figure, you can see that one complete 360-degree turn took around 3.4 nanometer through the double helix. Therefore, there are 10 base pairs per turn. The external diameter of a helix is always 2. The external diameter of a helix is 2 nanometer. That means the distance between two strands in a double helix is 2 nanometer. Because of the way the bases bond with each other, the two sugar phosphate backbones of the double helix are not equally spaced from one another along the helix axis. These unequal spacing results in groups of unequal size between the backbones. One group is called as a major or wider group, and the another one is the minor group. You can see from here, this is the major group, where here you can see the minor group. The edges of the base pairs are exposed in the group, and both groups are large enough to allow all the particular protein molecules to make contacts with the bases. Now, scientists have identified 3 different conformations of DNA double helix. That is, these are aDNA, bDNA, and zDNA. The most common conformation in most living cells, and which one is actually proposed by the Watson and Crick, is known as bDNA. Here you can see this is the bDNA. And this is the space-filling model of different conformers of the DNA. There are also two other conformations, that are aDNA, which one is a shorter and wider form that has been found in the dehydrated samples of DNA and rarely under normal physiological circumstances. Another one is the zDNA, where the right one you can see. The zDNA is a left-handed conformation. zDNA is a left-handed conformation. You can see that here from the figure, it is a left-handed conformation. zDNA is transient form, temporary form of DNA, only occasionally exist in response to certain types of biological activity. Scientists have discovered that certain proteins bind very strongly to zDNA, suggesting that zDNA plays an important biological role in protection against viral diseases. So this is a tabular format of all these 3 different components of DNA. You can see that bDNA is actually right-handed helix, aDNA is also right-handed helix, but zDNA is left-handed helix. The most abundant form of DNA in biological circumstance, that is a B form, where the A form is actually exist in very dehydrated conditions. The 3-dimensional arrangements are also a little bit different. bDNA has a very solid central core. Major groove is wider, deep, and minor groove is narrow and deep also. But in case of aDNA, the central core is hollow. Narrow and deep major groove is there, and wide and shallow minor groove is there. Okay, so there is a difference between all these among all these conformers regarding the helix diameter, helix speed, base pair per turn, helix twist, all these parameters. Now this session is up to this. These are the references. Thanks to all of you.